Good morning, everyone. I'm going to talk to you today about the work we've been doing on Amitraz resistance in Varroa. So Varroa is the major cause of colony losses. These are the self-reported causes of colony death as reported by the Bee-Informed Partnership. And as you can see, most of the colony losses are due to Varroa. However, I'm sure if you included losses due to poor winter, weak, CCD, don't know, and other, the actual causes would probably be Varroa, because as you can see here, Varroa are small and can't really be seen that well, but it's also the viruses that they transmit that cause colony losses. So viruses such as the deformed wing virus and chronic bee paralysis virus really wreak havoc on colonies. Due to those colony impacts, beekeepers often implement chemical control measures. So materials containing Amitraz, oxalic acid, formic acid, or thymol are typically used to control Varroa populations. However, over-reliance on these materials as the sole measure for Varroa control often can lead to resistance. And in fact, the first report of Amitraz resistance in Varroa in the United States was reported over 20 years ago. So in this project, they treated colonies in these apiaries with strips containing fluvalinate, coumaphos, or Amitraz, and then measured the change in Varroa population 6 weeks later. Colonies that were treated with fluvalinate strips, the Varroa populations increased 89%, indicating that those populations were resistant to fluvalinate. Coumaphos treated strips reduced populations 97%, which is a really good indication that they're susceptible to coumaphos. And Amitraz did reduce the population about 75%. However, this gap between 75 and 100% has been indicative of resistance. So they collected Varroa from those populations and performed the glass vial bioassay test, and we'll see the details of this later. Using different concentrations of Amitraz, they were able to determine the LD50 value, or the lethal dose that kills 50% of the population. In doing that calculation, you also come up with the LD90 dose that kills 90% of your population. And that's typically very good to use in resistance monitoring. So if you see 90% mortality susceptible, less than 90% indicates resistance. And in fact, that's what they found when they looked in the field. So Varroa from Texas had 85%, which is nearly 90% mortality, whereas the Varroa from Minnesota had significantly reduced mortality at that concentration, only 32%. So that's a pretty good indication of Amitraz resistance. However, in the intervening 20 years, not too many reports of Amitraz resistance have been confirmed in the field, but we did hear anecdotal reports from beekeepers that Amitraz doesn't work like it used to. So we wanted to put some numbers behind that, and we started this project in 2019. And the goals initially were just to determine the extent and degree of Amitraz resistance in commercial beekeeping operations, and then validate measurements of Amitraz resistance. So the first method that we used was the glass vial bioassay test. And these are small vials that are treated with a known concentration of pure Amitraz. So that way we know it's resistance to the active ingredient, not a misapplication or degraded material that's causing reduced control. So we treat those vials with different concentrations, place 10 Varroa with a white-eye pupae, and then measure the ones that are dead the following 24 hours, and we calculate our LC50 values to compare resistance between populations. We also implemented the APOVAR efficacy test, which is performed by putting about 300 bees in these cups that have a square of APOVAR in them, as you can see here at the top. After 3 hours, we count the number of Varroa that drop into this tray here, and we count that as number of Varroa in the tray. And then we wash the bees to dislodge the remaining mites, and we count that as the mites in the wash. We add the number in the tray to the total in the wash to get our total mites in the sample. We then divide the mites in the tray by the total number of mites in the sample to get our APOVAR efficacy. So if there's 10 in the tray, 10 total mites, 10 divided by 10 is 100%. However, if there's 5 in the tray and 10 total mites, that means our APOVAR efficacy is 50%. So this is a quick, easy way to measure Amitraz resistance. When we did this first initial survey, we actually had some good news in that 511 operations and 719 different apiaries didn't have enough Varroa to determine Amitraz resistance. So this is actually a good thing. So it's not widespread. So this is actually a good thing. However, when we did find resistance, it's restricted within operations and even within the apiary. So there's colony to colony variation. So in this graph, we have the results of the glass vial bioassay test from the 2019 data. So we have Amitraz LC50 on the left axis. And then on the y-axis, we have the different Varroa populations that were sampled. Lab is the USDA lab population that does not get treated with Amitraz, so it's susceptible. And then the different letters indicate different beekeeping operations, and the numbers indicate different apiaries within that operation. The resistance ratio, or RR, is simply the Amitraz LC50 standardized by the LC50 of the lab population. So this represents how many times higher the Amitraz LC50 is in that population. And so as you can see here, we found some Varroa populations that did not have any resistance. They were the same as the lab. Then we had low levels of resistance up to 5-fold. Between 5 and 10 was about intermediate levels, and then over 10 was high levels of resistance. Particularly in apiary B, in which these Varroa were actually treated with Amitraz, and we sampled them afterwards, and you can see the ones that we did collect had about a 22-fold resistance ratio. So high levels of resistance in the Amitraz viral bioassay test. In the apivar efficacy test, we saw something similar in which colonies had lower apivar efficacy, especially that colony B that I mentioned on the last one. Apiaries C and B had apivar efficacies that were significantly less than the USDA lab population, and so below 80% is our resistance definition. So you can see that's what's going on here. But you also notice that as apivar efficacy decreases, these error bars increase. So that's indicative of colony to colony variation. So some colonies are susceptible, some colonies are resistant even within the operation. So it's heterogeneous in the populations. Now you notice that B had the highest resistance ratio and also the lowest apivar efficacy. So if we plot those two factors against one another, actually what we do get is a very nice correlation in that the Amitraz resistance ratio, as that increases, so the apivar efficacy decreases. So this is a nice cause and effect relationship. So what's really nice is that allows us to use the apivar efficacy test as a resistance monitoring tool going forward, because it's much more simple to use and a lot more forgiving than the glass vial bioassay test. So this year we used a remote sampling protocol using the apivar efficacy kits to do our sampling remotely. So we sent these kits to cooperators up and down the East Coast, the United States. And then we provided detailed, streamlined, and video versions of the sampling protocol and data collection methods. This is very, very simple, and if you're interested, I could share those techniques later if you want to perform this on your own. Varroa samples and the data sheets were then shipped back to us here at the USDA, and in total, at the conclusion, we were able to sample 145 colonies from 16 apiaries and 11 beekeeping operations. So this is a pretty good sampling. So when we're looking at the apivar efficacy results this year, we did find that there's a higher degree and prevalence of reduced apivar efficacy in 2020. So again, the different letters indicating different beekeeping operations. And you can see here, they're all significantly less than the USDA lab population, which is 100% susceptible. But what was really interesting is that apiaries F and I, these were collected after the fall Amitraz treatment. And you can see here that apivar efficacy below 50% now in apiary F and 35% in apiary I are really indicative of the apivar efficacy that's going to lead to operational failure. So this might be very important to look at going forward, that we can use this apivar efficacy of less than 50% as our operational control measure. So what we can conclude from the surveys is that resistance has been detected, but its status is uncertain. Are we riding the wave of Amitraz resistance? Is this a new problem that's coming up that we need to pay attention to? Is this something that's stable? Is this something that's not really too much of a concern to some people? We need to do that with more resistance monitoring. The resistance varies among colonies, among operations. So this shows us that even when we do find resistance, it's a little bit like looking for a needle in the haystack, because some colonies might be incredibly resistant while adjacent colonies can be susceptible. So we have to understand what leads to that variation in the resistance phenotype within the operations. And what's really nice is that the apivar efficacy test is a reliable surrogate of the Amitraz LC50, so we could use that as a resistance monitoring tool going forward, which is really, really nice because it's simple, easy, and cheap to use. And all these results were published, from 2019 were published in this public reference cited here. So what we want to do in the future is to assess resistance at the colony level. What is the amount of reduced apivar efficacy that leads to operational control failure? It seems like this year we found out it's less than 50%, but we need to confirm that with more samples going forward. I'm not going to really base that on a sample size of 2 at this time, but it's something we need to do going forward. Understanding the biochemical mechanism of resistance, so whether it's enhanced detoxification or target site insensitivity, are really important for dictating management strategies going forward. Then we also need to look at how Amitraz is being used in these operations that might lead to resistance. So whether it's over applying in terms of frequency or intensity, we need to get at that so that way we could simply come up with easier control measures to mitigate resistance. And then we're going to work on identifying and developing genetic markers of resistance. And this is actually a project I'm currently working on with Ariane Avalos, the geneticist at our lab, with funding provided by the project APIS-M. So we're going to use the APIVAR efficacy test to find resistant and susceptible mites, then do whole genome sequencing on 300 samples, which is a huge effort. And then Ariane's going to use his skills to identify genes associated with Amitraz resistance, and then ultimately leading to the development of a molecular diagnostic test. And for beekeepers, what this is going to result in is the benefit of a low-cost, extensive, and rapid evaluation of Amitraz resistance. So hopefully in the future, you just ship us a sample of Varroa, we grind them up, look at the DNA, and tell you if they're resistant or not. So that way you can make management decisions going forward. And we really need to establish a resistance management network. So I envision this as a collaborative network of university scientists, government researchers, beekeepers, and apiary inspectors. So that way we can provide more consistent monitoring of Amitraz resistance across the country. So the more people looking, the better the data is. So that way we could head this off at the pass. Utilizing those same research groups, we could also understand the hows and whys of Amitraz resistance. So including population genetics, molecular biology, biochemistry, and pharmacology of Amitraz resistance is really important because that's going to ultimately help us develop practical management techniques to extend the utility of Amitraz and other miticides going forward. So we're always looking for people to help out with this resistance monitoring program. So if you're interested, let us know later. And I didn't do all this work by myself. It took a lot of really great beekeepers to help out, either giving us access to their colonies or actually doing the tests themselves and shipping the samples. So we're very thankful for that great collaborative environment. And then Victor and Dave, my two technicians, who helped me develop these protocols and collected samples in 2019 and shipped out materials in 2020. And with that, we'll take any questions.